Hello and welcome to the next A-level video from the conscientious biologist Ben Gallagher. This one's the second one in the nervous system playlist. This one's much more complicated and starts to add lots of A-level detail to the more basic and fundamental recap stuff that we did in nervous system 1. This is the bit that most A-level students struggle with. It's quite fiddly and complex understanding the action potential. So I've broken it down into really straightforward and highly visual steps. So hopefully by the end it will all make total sense. As always, please do subscribe to my YouTube channel if you haven't done so already. It really does help me out to have more subscribers, and check me out on Facebook and Instagram for lots of additional material and cool infographics. Thank you. So if we think about nervous communication, it works a bit differently to electrical communication in an electrical wire. Now you might assume it would work the same and that we have something that travels from point A to point B. Because if I want to touch something with my finger, then the sensation I feel here surely just needs to travel up to my brain to inform me about it. But it's not as simple as something traveling from point A to point B carrying a message. That is what happens in an electrical wire. You have actual electrons zooming down the wire. But instead of anything moving down a neuron, what you actually have is a wave of excitation. It works more like this, like a Mexican wave if you've ever done that in a football stadium or anything. So those yellow faces next to me here, none of them moved along from A to B. They just went up and down. They got excited and then went back to their original state, if you like. It was that wave that traveled along. That's how a nerve impulse works, by this wave of excitation. So what we need to study in this video is how a part of a neuron can get excited to pass that excitement on to the next bit and the next bit, and then return to its original state. That's the quite complex bit of this video, but I'll make it as straightforward as possible. So we need to recap membrane transport first, because to get that excitation, you've got to be able to move things back and forth through a membrane. So it's a quick review of year 12 material. This should look very familiar over here, that it's a phospholipid. Phospholipids, remember, have a glycerol head. They have fatty acid chains underneath, that the head is water loving, which makes it hydrophilic, that the tails are water hating, which is hydrophobic. And because of that, they will always arrange themselves in water in a very specific way. If there's not many of them, they might form a mycele, which you should remember is just where they sort of form a circle with all the tails protected on the inside. But biologically, we're interested in them forming lines like this with an opposing line facing them to keep all the tails on the inside, the heads on the outside, so the heads are near any water, the tails can be dry on the inside, and that's your phospholipid bilayer. If that's at all unfamiliar, go back to one of my previous A-level videos where I go through that in much more detail and we look at lipid biochemistry. But hopefully that's just a quick review. Now remember about how things travel through phospholipid bilayers. If they're going to go by ordinary diffusion, they can move directly through the phospholipid bilayer by just normal diffusion, and that's for small or non-charged particles, things like oxygen or carbon dioxide. Now if you've got large or charged particles, they can't go through the fatty acid chains, so they need to be helped, and they're helped by large proteins, because remember, proteins do pretty much everything. And these large proteins that are transmembrane will have a channel through them through which those particles can travel. But they still have to travel from a high concentration to a low concentration, so down a concentration gradient. But because they need to be helped, and the proper word for help is to facilitate, this is facilitated diffusion. And that's for large or charged particles such as glucose or sodium ions. Now we've talked already about a nerve impulse being an excitement. Now that's a charge, okay, a positive charge or a negative charge. So look, we've already got here, we're talking about charged particles. So you know facilitated diffusion's got something really important to do with nervous signaling. Now if we want to move things against the concentration gradient though, so taking the same picture here, but if we think of it in the other direction, if I want to move things from here at low concentration up to high concentration, I need a different kind of transmembrane protein, one with a little sort of mouth area, a binding site that your molecule can go into, that it can then be pulled into the molecule and pushed out the other side. Hopefully you're looking at that and in your head you're saying that that's active transport. Active transport is movement against the concentration gradient, and it requires energy from ATP. Now that's also required for setting the correct charges on a membrane for nervous signaling. So this is a quick recap. Go back and watch the video on membrane transport and lipid biochemistry if this isn't looking familiar, because you're not going to understand anything that we go on to if this doesn't all make total sense to you. Okay, right, let's start looking at how the membrane of the neurons control that charge to create that wave of excitation. So this is where it gets complicated, but stick with it. So we're talking about two ions here that act in the membranes of a neuron to create the charge. We've got sodium ions, which are Na+, and we've got potassium ions, which are K+. Now notice both of them are a plus, both are positive ions. It's important you remember they're both positive, but we're still going to make negatives from positives. I'll explain how. If I put on here our motor neuron that we've talked about before, just so you understand where I'm talking about here, I'm going to circle that specific node of Ranvier because that's where you can see exposed membrane. So if I magnify it in that box, you can see the ends of the two Schwann cells. You can see the black lines representing the membrane of the axon of the neuron. What I'm going to focus on next, if I circle in that yellow one and enlarge up there, we're talking about that section of membrane, phospholipid bilayer. But if I move that down to the bottom here and represent it just as that long black line, okay, so that black line over there is the cell membrane of the axon in the node of Ranvier of a neuron. To make that even more clear, what we're talking about is the pale blue stuff here is the cytoplasm inside the neuron, and the stuff above is the tissue fluid surrounding it. So you've got liquid on the inside of the cell, liquid on the outside of the cell. The membrane is a barrier between them. Now we need to achieve a voltage inside the neuron. That's what's going to allow us to create some excitation by positive charge and bring it back down into a negative charge. Now this is where I should explain that point as to how I'm going to make negatives out of positives. And it's all because voltages are known as potential differences, which is the difference between two parts. So imagine if you had 10 chocolate bars. Brilliant, you're positive, you've got 10 chocolate bars, plus 10. Imagine I've got 5. Hooray, I've still got 5 chocolate bars, I'm plus 5. But relative to you, I'm negative 5 because you've got 10, I've got 5. Yes, we're both positive, but I'm 5 less than you. So we can achieve negatives purely by looking at the balance between positives. And if one is less positive, we could count that as being negative because it's negative to the higher one. Hopefully that makes sense. It should do in a minute when we explain the process. So first thing that we need to do to have an action potential is we need to have the neuron at rest to start off with. That's like in the Mexican wave example, you're sat in your seat just waiting for the Mexican wave to come round to you. Okay, now resting potential is minus 70 millivolts. It's still positive in here, but what we mean is that it's 70 less than the outside. So it must be really positive on the outside of the cell and 70 less positive on the inside. That's what we mean when we say it's minus 70. Now that is achieved by having these proteins in the membrane. These are transmembrane proteins, and these ones are called sodium potassium pumps. Pump implying it must be active transport. Now these sodium potassium pumps are kicking out sodium and bringing in potassium. But of course, if you were just sending out one positive sodium, bringing one positive potassium, then the charge would stay balanced. One goes in, one goes out. So to achieve the negative on the inside, you must be sending more positives out than you're bringing in. And it's actually a 3:2 ratio. 3 sodiums are being sent out for every 2 potassiums coming in. That means there's more positives going out than you have inside. So it's minus 70. There's 70 less positives inside than outside. That should by now be making sense to you. That will maintain the resting potential of minus 70. which in itself makes a mockery of calling it a resting potential, because active transport requires energy. So to maintain rest, the membrane actually needs to be highly active to be pumping those neurons out, pumping those ions out, sorry. Now in pumping those out, what you're doing there is you're setting up some concentration gradients as well. You're putting all your sodium on the outside, so high concentration of sodium outside the cell, high concentration of potassium inside the cell. Now that means we can now move on to the action potential itself. The action potential is the excitement. That's the up bit of your Mexican wave, or on my previous slide where I showed the little yellow faces, it's the up bit. Okay. Now to achieve the action potential, the up of the Mexican wave, which is plus 40 millivolts, the positive, you're going from down at minus 70 up to plus 40. All that needs to happen is the sodium potassium pumps can stop. So you'll notice I've removed the arrows showing the sodium potassium moving, and you open a second channel, the green one there, which is a voltage gated sodium channel. If those sodium channels open and they're for facilitated diffusion, then sodium should come flooding in because of the concentration gradients you set up at resting potential. You put all the sodium on the outside. If you now open those channels, sodium's going to flood in and make the cytoplasm really positive and change that minus 70 to plus 40. That's your up. Okay. That's going to affect the next bit of neuron to make that one do the same thing, and so on. And that's how a Mexican wave continues. It's how my diagrams on the first slide showing the little yellow faces going up and down will continue. But we've now got to put this back, and putting it back needs repolarization. That was depolarization before, where you went from minus 70, you depolarized it to plus 40. We've now got to try and repolarize it to get it back down the other way. Now, of course, hopefully you might have noticed you've still got all your potassiums on the inside that you put there during resting potential. So all you really now need to do is to open other channels which can allow the potassium through. So these are voltage gated potassium channels, because that's going to allow your potassium to flood out. And if potassium floods out by diffusion, or by facilitated diffusion, I should say, that's going to turn the inside very negative, because you're losing all those positives to the outside. But it goes too negative, it goes to minus 85 millivolts, and you do need to know these voltages. You need to know the specific numbers. We call that minus 85 because it's gone too far polarized, hyperpolarization. But of course, to now correct that, you just need to reactivate the sodium potassium pumps and re-establish the resting potential. Because if you can move the sodium potassiums back to where they were, it should go back to where it started at minus 17 millivolts. Watch that again if you need to. Take screenshots after each one of these bits of writing completes so you can see it. But what I'm going to do, I'm going to show you something really quickly. This is something I've been teaching for years and years and years that I made up about 15 years ago. What you could do, if you write Na plus on your one hand and K plus on the other hand, then you can do something really easy to help this stick in your head. If you imagine sodium, so during resting potential, sodium got kicked out and potassium got pulled in. Okay, so if you imagine the neurons in front of you here, sodium's been kicked to the outside, potassium's down on the inside. That's resting potential. Action potential is the sodium coming in. Then we want to repolarize by kicking the potassium out. Then you need to switch them back. So it's resting potential, in, out, switch, in, out, switch. That's basically all that happens. Sodium, that's resting potential, has put these into the places that they're in. Sodium on the outside, potassium on the inside. Sodium comes in, potassium comes out, they reset to start position. In, out, switch back, in, out, switch back. Really simple. Sounds really complicated with everything that's on here, but as long as you understand the membrane and the transport proteins, it's literally sodium in to get your, that's your hooray, that's your Mexican wave positive. Potassium goes back out, reset them. In, out, reset. Simple. Take what screenshots you need to, but this is really, really important. What we're going to look at next is how those voltages look on a graph. So this is how they love to ask about the nerve impulse when it comes to the exam. But I'm going to show you this graph, and we're going to call this version 1, because in one of the next videos on saltatory conduction, there's a bit of extra stuff we need to add to this. We'll bring this exact graph up, the same descriptions, but with a little bit more. But this is how it applies to what we've just talked about. So if we've got a graph of a nerve impulse over here, I've got the voltage over on the far side in millivolts. I've got the time on the bottom in milliseconds. So you can see on this graph that we're only talking about a couple of milliseconds to get a nervous response, to get a reaction. Okay, if we put some values on this, you should be able to recognize that over on the far side there you've got -70 because it's at rest. The voltage is then going to go up high to +40. That's at the top of your peak. You're then going to move this potassium back out to take you back past 0 and all the way down to -85. Then you're going to kick out the sodium and potassium to go back up. So if I put some letters on there, A to F, we need to explain each one of those points. So A, we know, is the sodium potassium pumps actively transporting out 3 sodiums for every 2 potassium ions. That's 3 sodiums out for potassiums coming in, making the outside more positive than the inside, setting up those concentration gradients that we're going to need, the depolarization to action potential and the repolarizing down to hyperpolarizing. That's the resting potential, that's -70. Now B, we're going to skip for now because that's what comes up in the next video. Don't worry about B. All I want you to worry about here, where B is that's kind of the start of the action potential, but there's a little bit extra that happens at B. So we're going to jump straight on to C, where we've got the current, the voltage increasing because those sodium channels have opened. Sodium's going to come flooding in. That's going to raise us up to +40. So influx of sodium, influx means it's coming in, and that's only because the voltage hit a correct level for the voltage gated sodium channels to open to allow that sodium to come in. But of course, once they get to D, which is the action potential of +40, once you get to that top point, that voltage of +40 closes the voltage gated sodium channels. The voltage gated bit just means they respond to certain voltages. Well, they respond to the +40 by closing the sodium channels and opening the potassium channels. That means at E, you're going to get an efflux of potassium. Potassium is going to leave by facilitated diffusion, which is going to decrease the voltage in the neuron cytoplasm, which is why the graph comes absolutely crashing down until you get to F, which is the hyperpolarization of -85, where the potassium channels close at -85 and the sodium potassium pumps reactivate, start kicking out all the sodium that flooded in at C, to start pulling back in all the potassium that flooded out at E, to get things back where they should be, which takes us back up to A. So this just repeats round and round. Now in the exam, they're likely to give you that graph. They could put an X anywhere on the graph and say, explain what's happening at X, but it will be one of these. The only bit to add is what's happening at B, which we'll look at in the next video. So head straight to that video. There should be a link for it appearing on this page. That's Nervous System 3 saltatory conduction and threshold potentials. That explains how the signal is passed from person to person, because in a Mexican wave, you can see the person next to you going up. So you know when to go up. There's no eyes within a neuron that can see that the last bit has become positive. So there must be some kind of signal between the positive jump up the action potential here and how that's communicated to the next one. That's what we're looking at in the next video. So head there next, please. Please do give this video a like if you find it useful. Recommend it to your friends. Send it on to your biology teachers. I'd love this to get used as widely as possible. If you can, please do subscribe to my YouTube channel if you haven't done so already, and go and have a look at all my information that's on Facebook and Instagram. Thank you.